Yes. Well, I'd also like to start by thanking the organisers for giving me an opportunity to go from A to B, which seems to be how to get to a restaurant. A was in Sydney and B is in Berlin, and it was quite a pleasure to be able to leave sunny Sydney and arrive in sunny Berlin, so thank you. So today I'm going to talk to you about our ongoing work on understanding epigenetic remodeling in cancer, and then go on to talk about our unpublished work on what impact this might have on changing replication timing also in cancer. So as we all know, cancer is a disease of both the genome and the epigenome. So in addition to the genetic changes, which involve specific mutations, deletions and amplification in key genes that are associated with carcinogenesis pathways, as described here in the Hanahan and Weinberg cell paper, there's also a myriad of epigenetic changes, including changes in both hypermethylation at CPG islands and hypomethylation in intergenic regions. And this is in addition to the myriad of histone modifications that occurs also at both single genes, and as we now know from our work and many other people's work, these epigenetic changes also can encompass large domains across the cancer genome, similar to large domains of deletions and amplification that can occur genetically. So one of the challenges that we have had in the field over many years is to map both the genetic changes as well as the cancer epigenomes, so that we can understand what the relation is between the genome and the epigenome. So we've been doing this for a number of years, and today I'm going to talk to you about our ongoing questions of cancer regional domains. And so first of all, we've been looking at mapping where regional changes occur in cancer, both long-range epigenetic silencing, we've termed LRES regions, as well as long-range epigenetic activation, we've called LREA regions. I'm going to talk to you about what we're discovering about common epigenetic features that underpin these regional domains, and then lastly go on and talk to you about the impact of these changes potentially in replication timing in the cancer. So as I said, we've been putting together epigenome maps now for quite a while in prostate cancer, and this has been the work of primarily of Claire Sturziker and the bioinformatician Aaron to analyse the data. And so between normal prostate cells and prostate cancer cells, we've been combining low sequencing of input DNA and SNP arrays to look at the DNA sequence. To look at methylation, we've been doing 450K arrays, MBD-cap-seq, and whole genome bisulfite sequencing. And histone modifications, we've been doing ChIP-seq and GNOME-seq to look at nucleosome positioning. To look at transcription, we've been doing directional RNA-seq and CAGE-seq to look at promoter usage, and all of this to really understand what it is about the combination of these various marks at the DNA level that prescribes whether regions are going to be active, as in long-range epigenetic activation, or repressive, as here in the TROPS, that dictates whether it's going to be a region of long-range epigenetic silencing. So also to understand regional deregulation and replication timing, we've used Replication-seq also in these normal and prostate cancer cells. So today I'm just going to give you a summary of what we now understand by compiling all of these epigenome maps together. So where are these changes? And so to first of all look at where these epigenetic deregulation domains were in prostate cancer, we took an approach where we first looked for changes of gene expression. And we looked in the public database at the time. In Oncomine, there were 215 normal gene expression profiles and 380 prostate tumour profiles. And we combined this with prostate cancer cell line expression profiles. And Mark Robinson, a statistician in the lab at the time, developed a T-statistics tool to basically do a sliding window walk to look for domains that were commonly deregulated in prostate cancer. And so we took an arbitrary T-statistic over 5, so it was highly significant, and looking at regions of change. And in this analysis, we looked for regions where we had a median of 4 genes that were deregulated in the same region, either activated or silenced, and we excluded regions that had undergone either deletions or amplification. And this just shows you a typical output where we were looking for contiguous changes in gene expression, and this is an example of an area in prostate cancer that is commonly activated. So we found many of these regions, and mostly they were in discrete places in the genome, but plenty of them were like this gene family, the kallikrein gene family, where in prostate cancer we had a domain of activation adjacent to a domain of gene silencing. So in using these parameters, we identified in prostate cancer 47 regions of silencing and 36 regions that showed activation. So the next question we want to know, of these regions, what were the underlying epigenetic changes associated with this change in gene expression? So to do this, we combined the chip data with the DNA methylation data, and here I'm just showing you the output from the start of transcription, where we're looking at windows at about 1 kilobase and 2 kilobases upstream from the start of transcription. And this is where we've got the contiguous genes going down here, and this is the change in the chromatin marks between the normal cells and prostate cancer cells. And this just shows you 2 of these regions, and as you can see very strikingly, we find that the contiguous genes here that are associated with gene repression are showing a loss of the active histone mark H3K9 acetylation here and here. But in addition to the loss of the active mark, we also find that there's a contiguous gain of repressive mark, and here you can see in red the gain in cancer of H3K27 trimethylation, and commonly gains in DNA methylation. We found across the genome that these domains commonly occur in regions that are about 1 megabase in size. But in saying that, these domains have different borders, and we still don't really understand what determines where the borders start and when they finish, depending on the chromatin modification that we're looking at. And you can see here, though, even though there's commonly a loss of histone active marks, there's commonly a gain of repressive marks, and we also see an exchange of repressive marks that has been reported a lot in cancer, where you have a loss of the H3K27 and a gain of methylation. But as you can see, that's not the only change we get. We also get regions where both K27 and methylation can co-occur. So there's a multiple range of modes that we identified associated with these regional changes of expression. When we looked at the epigenetic changes underpinning gene activation, we also found that these occurred in a similar size region of around about 1 megabase in size. And again, when we looked at the changes around the transcriptional start site, we found that these occurred also in blocks. And for gene activation, we found the opposite of gene repression. Here we find that there's an increase in the active marks, H3K9 acetylation, an increase in H3K4 trimethylation, and in some of the regions, a decrease in the repressive mark H3K27. So in many ways it's using a similar mode of change, but in the opposite direction. So for the regions of activation, we were very keen to see whether we could see any evidence of DNA demethylation, because even though it's clear in cancer, hypermethylation of CpG islands is very commonly associated with gene repression. The dogma is that hypomethylation or demethylation of CpG promoters might be a way of activating, for example, oncogenes. But we wanted to do this systematically to see what evidence there is for demethylation in cancer and gene activation. So to do this, we used at the time our MBD-CAP-seq data, and again we looked at methylation changes at the transcription start site plus or minus 2.5 kb. And we correlated this with gene activation. So what I'm showing you here is a change between normal and cancer, where we've got a change in DNA methylation and a change in activation. So in the long-range epigenetically activated regions, there was 135 CpG islands, and we were very surprised to find that these genes associated with these CpG islands, they're all activated as we know, but what we were surprised about was that the majority of them increased in methylation. So 23% in this quadrant gained methylation. Many of these islands didn't change their methylation status because they're already unmethylated. We were predicting that gene activation would be correlated with DNA hypomethylation, and in fact, we only had one of these CpG islands here that was hypomethylated and activated, which was really quite a dilemma at the time for us to understand what was going on, because it was against what we were predicting, what the dogma would have told us. So we looked more closely, and this CpG island that showed hypomethylation was associated with kallikrein 4. But when we looked at the kallikrein 4 locus, we found that there was 2 CpG islands associated with kallikrein 4, one losing methylation and one gaining methylation. So to look at this in a little bit more detail, this is the kallikrein 4 gene. These are the 2 CpG islands. This is the methylation data from normal prostate cells, and in this particular case, Allen-CAP cancer cells. And as you can see here, there clearly is a loss of methylation, which we validated with bisulfite clonal sequencing. But when we look at the upstream CpG island, this upstream CpG island had indeed gained methylation, and it was this combination that was associated with activation of kallikrein 4. So how did this, what was the mechanism for this? So we looked more deeply at our data, and again, this is the kallikrein 4 locus, these are the 2 CpG islands. We looked at our RNA-seq data and our CAGE data. Now the CAGE data, which is telling you where the start of transcription is, and the RNA-seq data was telling us that in cancer, rather than starting at the canonical promoter in the first exon, transcription was actually happening at the downstream exon. And this activation in cancer was actually associated with this upstream hypermethylation, but a gain of the active mark. And so this was an example where this differential methylation was actually changing promoter usage and hence leading to activation of this transcript in cancer. So that explained this one gene. So then we thought, well, what is happening with all the rest of these CpG islands that gained methylation and gained gene activation? So we looked at all of these CpG islands, and we could class them into 2 classes. The first group were CpG islands that gained extensive hypermethylation at the promoter CpG island, and this is just showing you an example of this, which is like the kallikrein 4 locus. This is the TRIM36 gene. Again, this gene has 2 CpG islands, one upstream and one in the middle of the transcript. And here we have, this is the gain of methylation across the whole of the CpG island. And this gain of methylation is associated with increased transcription, again of this H3K4 trimethylation mark. And here we can see that a change of the promoter usage, because this is where the canonical promoter would be, and here we've got transcripts in the cancer coming downstream, being triggered on this internal CPG site. And so these gains of methylation in these extensive group of CPG islands that were hypermethylated correlated with a change in promoter usage and a change in the transcriptional start site. The next group of hypermethylation that we found associated with gene activation was a little bit more intriguing and something that we're still trying to understand. And this is where, rather than extensive hypermethylation across the entire CPG island, we find that methylation is encroaching in the borders of the CPG island, but doesn't span the transcriptional start site. And again, there's an increase in the H3K4 trimethylation. So it's this combination. It's not changing where the start of transcription is, but it's promoting and allowing ectopic expression of a transcript, in this particular case, which is not normally expressed in prostate epithelial cells. So if we combine all of these CPG island hypermethylation, and as I said, we could divide them into two different groups, the majority of them, 65%, belong to this border methylation class, where we've got an encroachment of methylation at the borders of the CPG island. And 25% of them were associated with a change of promoter usage. So this clearly, in our minds anyway, shows that demethylation in cancer, at least of CPG island genes, is not very common. In this case, as I said, we only found one example. The rest of them were hypermethylation was associated with a change in either transcript usage or ectopic expression. So then we've now gone on to try and understand how in the same cell you can have regional domains that lead to activation, regional domains that lead to silencing. And so there's a number of hypotheses that we're addressing. One is that these regions actually change their localization, organization in the nucleus, so spatial changes and changing their association with the nuclear lamin. Another is that they're also changing their structural status and changing their association with CTCF and or cohesin deregulation. And the third one that we're exploring as well is that these changes in the histone changes and the DNA methylation changes impact on replication timing in the cancer cell. So today, just in my remaining time, I'm going to talk to you about our new work on replication timing in these cancer cells. So just to remind everybody, replication is a highly programmed process that occurs in each cell, and a gene will always replicate at the same time within S phase. And there's multiple initiation sites that occur, and this is important to ensure that there's complete replication during the S phase. And so in mammals at least, there's been estimated nearly 250,000 of potential origins of replication. And these occur in domains, 4,000 to 5,000 domains across the genome. And these domain sizes are up to a megabase in size. And it's really important, I think sometimes it always amazes me, that the cell only takes 8 to 10 hours to actually replicate, which is why we need so many multiple sparks of replication across the genome. So when we look at DNA replication, and as I said, as we all know, a cell replicates once per cell cycle in the S phase, it's not only the DNA sequence that has to be replicated in that 10 to 12 hour period of time, it's also the entire epigenome that has to be replicated to ensure that the expression profile is maintained. And so this means the DNA methylation profile has to be replicated, the myriad of histone modifications, the nucleosome positioning that we heard about from Peter, and this all makes up the nuclear architecture. And it's very important that all of this occurs, as I said, during replication, so that the daughter cells have the same transcriptional memory in the somatic cell system. So David Gilbert has been working in this field for a while, and there's this lovely photograph that he just recently published in Cell. And this is a lovely way to represent where the replication domains are in the cell. And you can see here he's stained the different replication foci, and the early replicating regions, which are shown here in green, are associated with the nuclear interior of the cell, whereas the late replicating regions here, are stained in red, are associated with the nuclear lamin. So what's interesting about this demarcation of replication timing is that the domains that replicate early in the S phase are commonly, they're gene dense, they're highly expressing genes, and they're associated with active and open chromatin. Whereas the domains that replicate late in S phase are commonly gene poor, they are associated with the nuclear lamin, and again associated with repressive chromatin. So this was very similar, we've thought, to the domains that we were identifying in cancer. So as I described earlier, we found that there were these domains that had either repressive or active marks associated with a change in their gene expression. And so one of the questions is, why is there early and late replication in a cell? And so one possibility is that it's this time difference that allows the epigenome to get its flavour put on, depending whether it's early or late. So early associated with active marks, and late associated with repressive marks. So I'm now going to just talk to you about how we've now addressed whether we can see whether our domains we've seen in cancer are actually changing their replication timing. And so to do this, Saul Burt, who is a PhD student in the lab, developed replication sequencing. And to do this, he used normal PREC prostate cells and compared them to prostate cancer Allen-Cap cells by BRDU pulse labeling, and then sorting these cells into 6 fractions of the cell cycle. And this just shows you one of the sorts of PREC, where we can sort G1, 4 S phase fractions, and 1 G2. And then using immunoprecipitation and high throughput sequencing, we were then able to visualize the replication sequencing after smoothing and mapping to the genome. And so this just shows you an example of replication sequencing to give you a feel for it. So these are the 6 phases that we sorted, and this shows you where early replication is in the green here. And as you can see, it changes from early to late replication here. Now, this data we've done in duplicate, and then we've smoothed the data so that you can actually easily see how it goes from early, late, early, late, early, late, all across the genome. So the first thing, when we compared the normal cells with a cancer cell, we were really very surprised how remarkably similar the replication pattern was, considering these cells really are quite different in their genomic architecture. And this just shows you a plot here between the normal and the cancer. And if you just look at the weighted average, you can see that there's this remarkable pattern of replication timing that is similar between the 2 cell lines. If we put all of this data together, and I'm sure you can't really see that, green is normal and red is the cancer, but just visually you can see how remarkably similar these patterns are. In fact, 95% of the 2 genomes, the normal and the cancer genome, had a constant replication time between the cells, and I really find that's quite remarkable. So this highlights the general maintenance of the replication regulation. But clearly we were interested in what was different, not what was the same. So the question we had is, in that 5% that was different, where were these differences located? So to identify the domains in the genome where the replication significantly changed between Allen-Cap and PREC cells, we located all the loci that had altered replication above a significant cutoff, and using this fairly high cutoff, we identified over 300 domains that replicated later, and that constituted about 3% of the cancer genome. And we identified 388 that replicated earlier. And these domains were reasonably small, 260 and 190 kb. So it begged the question, where were these sequences that changed in the replication timing? Do they actually harbour our identified long-range epigenetically silenced or long-range epigenetically activated genes? So the quick answer is we were really very excited to see that there was an amazing correlation. So here's an example of a long-range epigenetically activated region. And here in the normal cell, you can see that this region replicates late, whereas in the cancer cell, it replicates early. And this can be clearly seen here with the smooth data. Likewise, just showing you one example of a long-range epigenetically silenced region. In normal cells, it replicates early, whereas in the cancer cell, it replicates late. And again, you can see here this dramatic shift in replication timing. When we look at all of the defined domains, be they the active domains or the silenced domains, and compare that with the change in replication timing between the normal and weak cancer, we see that there is a significant trend for a change in replication timing across both the active regions that tend to replicate early and the silenced regions that are now tending to replicate later in the cancer cell. So we wanted to see how the methylation pattern actually correlated with this change in replication domains in cancer. So to do this, we looked basically at the change in replication timing. So here are genes that replicate early, here are genes that replicate late. A significant cutoff is either plus 20 or in minus 20. And these just highlights the genes that change. And what was interesting here, if we just look at the CPG island-associated genes, we find that whether they replicate early or whether they replicate late in the cancer, they still gain methylation. And this is summarised here, that whether they go into the late replication domain or the early replication domain, it's associated with CPG hypermethylation. But this now makes sense to us because we've already shown that hypermethylation is associated with gene silencing, but it's also associated with gene activation. So we then went on to look at at low-density CPG promoters, because CPG islands appear to be controlled in their methylation differently to low-density CPG regions. So to look at non-CPG island methylation, we used our whole genome bisulfite sequencing data, and again looked at the differences in the normal and cancer cells. And this shows you in the normal PREC cells, we're here looking at late replication over this side, or genes that replicate early over this side, and this is looking at the percent methylation, and this can be summarised here. So basically, in a normal prostate cell, we find that early replicating loci are associated with high methylation of these non-CPG island regions, whereas late replicating loci are associated with less dense methylation in the normal cells. But when we compare this with the cancer cells, we see a dramatic change, because in cancer there's a global hypermethylation, and this can be seen here in the whole genome bisulfite sequencing data. And summarised here, we can see that the early replicating loci in cancer is still associated with a high level of methylation. But what is really very dramatic is that the genes that change to late replication are the ones that have become essentially hypomethylated. So it's that body of hypomethylation in the cancer cell that we know about. So late replication in cancer is essentially unmethylated. So the conclusions I just will summarise again is that we're very surprised, I think, to see how remarkably well-timed the replication programming is between the normal and cancer cells. But where it did change, there was a correlation with these regional changes that we found in cancer, be it activation or silencing in the replication timing domains. We found that there was a clear association of hypermethylation in the cancer with a change to late replication DNA in the cancer cell. And we've gone on to now look at addressing the hypothesis that it's this hypermethylation that leads to a increased probability of translocations in the cancer, because in many of these hypermethylated regions are where we find common hotspots from the ICGC prostate cancer data. So that's it. I would like to just acknowledge my team in Sydney, in particular Saul, who I talked about the replication timing work, and Nicola, who's the statistician in the lab who did the analysis of the replication timing, and Claire Sturziker and Jenny Song have been involved in setting up all of these epigenetic maps that we've been working on. So, thank you.
Wonderful, thank you very much. So we're open for questions.
Yeah. What do you think is the mechanistic relationship between expression, transcription, say, and methylation in those few regions which change both transcription?
You mean what comes first?
Yeah, what comes first?
I would love to know. It's always the chicken.
What do you think?
That's right. It's the chicken and egg answer. But I think our studies to date are suggesting that the methylation is subsequent to the change in the transcription. Whether that's going to be a gene-by-gene basis, I don't know, but certainly of the specific examples we've looked at, it appeared that the methylation was a subsequent change. But it's early days, I think, to know generally.
I would like to ask a question as well. So you started off basically by an arbitrary choice of 5, and you took 4 out of 5, and now you end up at topological domains, whatever you want to call them.
Yes.
If you now turn around your whole thing, are these LRES and LRE regions really mapping over this entire topological domain, and how do you explain 180, 100, 200 kb fragments versus 1 megabase?
Yes, so look, I agree. I mean, it's an arbitrary cutoff because we had to do that to start with, and we've now changed our different cutoffs. But what's interesting in the replication domains that we're finding that are different are actually these smaller regions. Mm-hmm. And so some of our small domains overlap beautifully. Some of them are within the change, but not necessarily across the whole change. Mm-hmm. That's right. So we, as you may or may not remember, we did the expression analysis from clinical samples, and now we've gone back to cell lines. And so I think what we're seeing potentially is the heterogeneity, as we know, in clinical samples, and we're looking at these common changes in cancer cells.
So does it look more like a high C structure now, or are you doing a high C for that?
Yes, so we've done the high C, and we're challenged with the analysis of that at the moment. But the one thing I can tell you is the replication domains look very much like a high C footprint. Yeah. So it's really, the concordance looks like it's going to be very good.
Okay.
Oh?
Hello.
When you see the different, sorry. When you see this difference in the transcription activation of these hotspots, because you previously mentioned about the possibility of nuclear localization that could be influencing that, did you start to check where they are located into the nucleus towards specific landmark?
Yeah, so we're hoping the high C data would be able to tell us specifically whether it's changed the nuclear localization. So that's not something we know yet. But I'm also interested in one of the talks that we just had before, is that we've done a bioinformatic analysis and found an increased density of specific binding protein sites in these regions that are hypermethylated and associated with gene activation. So we're looking now to see whether it could be that the methylation actually is augmenting transcription because it's binding activators. But we'll see.
Okay, I think we have to move on, not to get out of schedule.